Good morning to one and all. This is Lalita Priyanka. Today I am going to talk about plant tissue culture techniques. So when you talk about the plant tissue culture techniques, it completely deals with in vitro cultures, like in laboratory cultures. So in general, we have a mechanism called as growing of a plant. For example, sowing a seed in the soil, and we are aiming to germinate that one. So that is a general criteria in the XYO. But the in vitro is not that much of simple. Here it requires some kind of aseptic conditions. So this particular plant tissue cultures or plant, what do you call is plant in vitro methods, culturing of plant in vitro methods. So generally, the criteria was first taken up by Herbert Land in the year of 1910. These studies are called as like laboratory techniques or else some kind of artificial mechanisms. So where you are going to stress on plant tissue culture is a collection of techniques used to maintain and allowing the plant to grow with the help of some kind of sterile conditions and some amount of nutrient culture mediums and known as MS media compositions like B5 media compositions. And this plant tissues cultures are widely used to produce the same clones, same as mother as the plant. And this method is also called as micropropagation, where we are aiming to get two to two types as the mother as the parental characters. The characters which are present in the mother plant, we are aiming the same characters in the daughter plants. So here the criteria is just maintaining the sterile conditions and having a good nutrient medium. In general, we used to have this MS media for culturing the plant tissues in the in vitro laboratory conditions, and this MS stands for Jurassic Cooks media, which is already designed media, and it comprises micronutrients, macronutrients, vitamins, some amount of salts like calcium chlorides, potassium iodide, and agar and agar. Agar and agar acts as a gelatin agent, gel. So this agar and agar mainly, when you add the agar and agar, so that gives the solid structure of the media. If not, if you are not adding the agar and agar media, so that only called as liquid media, or sometimes the liquid media is also called as liquid broth or liquid solution, right? This is not that much of suitable to a plant to grow. Why? Because in order to isolate the protoplast or any other thing, we require some kind of liquid media or broth. But if you want to aim these solid structures as callus, shoot and root organs like organogenesis and somatoembryogenesis, so we require a solid nutrient media. So these are simple techniques we use in the plant tissue culture laboratory. So we generally takes the explants. Why? Because this explants acts as your part of inoculation. So this explant is generally we dissect from the plant, and we used to sterilize the explant, and finally we place it on the nutrient medium and we allow the plant to grow. So from that explants we are expecting callus and shoot organogenesis and roots, some kind of roots and shoots going to be arise. So explant is one that nothing but any part of the plant taken out and grown in a test tube under sterile conditions in special nutrient medium is called as explant, right? So here why I am stressing on this explant is plants itself has a totipotent in nature. We know that plants they have totipotent in nature and we are pluripotent. Plants why they are having totipotency? For example, if you take any part of the plant, so they acts as an explant, and if you culture in the synthetic media or else this artificial, or we can call it as nutrient media, which is chemically synthesized in the laboratories, so they allow the plant to grow. They have the characters for allowing them to grow with the help of some kind of plant growth hormones, plant like auxins, gibberellins, cytokinins, ethylene in that way. We have plant growth hormones, and along with that some amount of vitamins, right? They also responsible the growth of the particular plant. So they are having they are totipotent and we are pluripotent in nature. We don't, for example, if I dissect my cell, any kind of cell or a tissue, something from my hand, if I place it on a media, the same can't be propagated, right? So this is not happens with regarding of higher organisms like human beings or anything. We are not completely differ from that. But plant, it has a criteria to develop into a plantlet. So this is called as explant. So we can take any part of the plant, so that acts as an explant in vitro conditions, not in ex vivo. So that you have to know. Next, methods. So if you want to do this particular plant tissue culture, we have to know some methods. That is type A, in the sense type 1 or type A or B, the in vitro growth of callus or suspension cultures. Callus is one particular term. It is also known as undifferentiated mass of cells. So if you want to see the image of callus, I will show you how this callus will be. And this is completely undifferentiated mass of cells. So this is the image of the callus, where you can't differentiate into root, shoot, stem or any organs like this. So this is completely this undifferentiated scum like this. You can't differentiate. It is light green in color, yellowish in color. Sometimes it is also in dark brown in color. So this, after 4 to 5 weeks or 7 weeks, then slightly they can have this apical growth like shoot and roots formations. Beforely, you can't identify the structure. You can't differentiate this structure. This is called as callus. The image of callus is like this, right? Next, right. So this is callus and suspension cultures where we are going to see in the liquid medium, broth. You have to know about the broth and the agar medium. Agar medium is a solidified nutrient medium where you will have the solid structure like in the earlier we have seen the solid structures like this. So this is the agar nutrient medium where we have agar and agar in this, and this has a solid gel-like structure. If I talk about nutrient broth, they don't have agar and they are liquid in nature. They are mainly useful for suspension cell cultures. Next, we have B type explants. Within the explants, we have one more criteria, that is single cell cultures, shoot cultures, root cultures, somatic embryos, meristem cultures, anther cultures, haploid production, protoplast culture, and somatic hybridization, embryo culture, ovule culture, and ovary cultures. So the all are acts as explants. Let me give single cell protein. This is single cell culture. Shoots, if you want to develop shoot the apical region, so that we can excise the explant from the shoot tips. And roots, you can dissect the root and again you can have it as an explant. Embryos, somatic embryos, especially from the corn or pisum pea or any other thing, we can dissect the embryo and we can culture that one. Apical meristem or else meristems that which are apically, and here the node and internode in between that we have an apical meristem. So you can easily dissect the meristems, the young part which are going to be just developing. So you can dissect that from the plant and we can put it on the medium. Next, anther cultures, especially datura flower, we can have this anther like structures. We can dissect the anther and we can take that one. Haploid production, this is also good examples when we have this haploid gametes H, N and N type single end. Protoplast culture, this is known as cell without a cell wall. They are naked cells. The easily we can isolate the protoplast from the plasma lemma. Next, somatic hybridization, where we have also talked about some kind of hybrid productions, synthetic seeds, that encapsulated seeds. So all these are examples for that. Next, embryo culture. So we can dissect the embryo and we can culture that one. Ovule culture, ovary cultures like androecium, gynoecium, what we have studied. So all these are acts as explants. Next, so we have to keep it in our mind when we are doing the experiment like plant tissue culture experiments. So environmental conditions also matters. There are three important aspects in the in vitro. How the ex vivo, how this ex vivo plant is growing in general. So it has an adaptations of tolerance to heat, to biotic and abiotic stress, like animals, if gazing animals come and eat, they preach their leaves, or else sometimes there is a unfortunate floods, or sometimes there is a drought conditions, they don't scarcity of water. Sometimes they may also have high temperatures, uh, like 60 degrees centigrades like that. Even though in that extreme conditions, extreme salines, extreme metal degradations, the plant sustain in ex vivo conditions. I'm talking about the ex vivo. But in vitro is not like that. They are completely sensitive, and they have to have this indications that nutrient media and some kind of sterile aseptic conditions and good aeration, good light, good photoperiodism. Everything should be maintained in the in vitro, right? So firstly we will talk about the nutrient media. Why? Because how we are eating the food, how we are sustaining our life, in the same way plant also requires some amount of micro macronutrients, amino acids, carbohydrates, vitamins, inorganic salts like some growth regulators, plant growth hormones, and optimum pH. So these are the criteria of a plant in order to develop in the in vitro conditions, in the presence of a nutrient synthetic media. So this media depends upon the type of the plant and the cell what we are going to culture. Generally, they require this both inorganic salts like macro and micro elements like potassium iodide, calcium iodide, cupric acid, cobalt chloride, magnesium sulfide, manganese chloride. In that way, so we have micro as well as macro units. Next, carbon source. The best carbon source for the in vitro cultures is sucrose. We have to maintain 2% and 3%. If you want to culture the media, 2% nothing but 2 grams per 1000 ml of media, nutrient media. 3% means simultaneously 3 grams, 30 grams to 100, 1000 ml of media. So vitamins, so we have to have add the vitamins also like nicotinic acid, thiamine, pyridoxine, oxins like this. Next, amino acids, especially arginine and methionine sometimes. They are also added if there is any requirement, then only. If not, if no problem. Next, growth regulators, especially auxins, gibberellin, cytokinins, ethylene. So they are going to be used for good cell and elongation mechanisms, apical root bud, lateral burst formations. And we have to maintain the pH like 5.6 to 5.8 and average is 5.7. It's very important for that. Next, aseptic conditions. Upon okay, this is that is with regarding of media. Next, we are going to talk about the aseptic conditions. So here they have to maintain some sterile conditions. The nutrient media should contain sugar, and by because it has 2%, as I said, 2% and 3% of sugars, where 20 and 30 grams of sucrose is going to be utilized. There is a chance of microbial growth. In order to overcome such problems, we have to maintain the aseptic conditions, right? So these are the microbes, they completely come in contact with the media. So the media get disturbed as well as your plantlet is going to be destroyed. Within no time, we have to discard that one. If we find any kind of bacterial or fungal infections with regarding of your tissue cultures, then priorly dissect that, but the discard that particular material. Then only we'll have the chances of having a healthy plants. If not, this plant affects the other one. Next, we have to take the importance of aseptic conditions, maintaining the temperature and the temperature as well as the photoperiodism light. So we have to give 16 hours of light and the remaining 8 hours of dark incubation. Next, we have to maintain the temperature here called 25 degrees plus or minus 2, sometimes 23 to 27, not more than that. So we have to maintain the optimum temperatures. And pH we have already known about 5.6 to 5.8 is an average, 5.7 we have to maintain. Next, sterilization is very important to stop the microbial growth. There is a high chances of infection for this particular in vitro plants. We have to keep mind that they should not be affected. Next, good aeration. So proper aeration should be cultured on the tissue culture, and which is very it should be spacious. It should be not that much of spacious and not that much of a very less in space, but you have to maintain the moderate level. Next, so they should where you are going to have these cultures, everything that should be cleaned, that should be maintained very hygiene and good proper what you are autoclaving, what you are doing in the laminar airflow, how the test tubes will be, how this particular orbital shakers or any kind of atomic shakers when we are using that one. Priorly we have to know about the equipment again. So here we have types of plant tissue cultures. We have different types how in the laboratory. The priorly we are going to have these callus cultures. So when you add this particular 2DZ or any kind of auxins like BAP, benzylaminopurine, so we are aiming this particular callus formation. So within the inoculation period, from inoculation period to 2 to 3 weeks, just we are going to have this particular callus structures. Callus is an undifferentiated mass of cells where it is slightly grown on upon the media. The coloration is light green in color to yellowish in color. If I talk about the suspension cultures, yes, these are the suspension cultures. They're the liquid cultures, liquid medium, right? These are called as suspension cultures. So we can dissect some amount of callus, not completely, just a small portion of this callus, and just add in the liquid agar medium. So this is agar medium, this grows in that way. If you want to add this callus in the liquid medium, so now this is a liquid medium where your callus is just placed here. After some time upon orbital shaker, if you want to dissect, so we are going to have this multiple cell clumps we can call it as. If you go with the mechanical, with the help of needle or any other kind of forceps, they may damage the cells. So this is the way we can easily isolate the pure lines of cells, right, in the liquid medium. So finally we isolate them, and one in the test tubes like this. For example, here we have nearly 7 to 8 different types of cells. So upon this one and one, we can add in the 7 different test tubes. So finally, we are aiming the callus from the single space. We are aiming the shoots and roots can be easily developed upon the hormones. What are the hormones? Growth hormones you are giving. So the plant responds accordingly. Next, maintenance. So this is the thing. This is the actual image what I have done in my MSc. So the real image, right? With the help of medicinal plants. Here we have taken Bacopa Manir, I think so. And this is the image how we have incorporated. After the 4 to 5 weeks of inoculation, we are just aiming the shoots. Clear? After the sufficient amount of callus grown, then we can shift the media into the subculturing. So this is also one of the example for subculturing, where from we have just cultured in after 2 to 3 weeks, then nearly 4 to 5 weeks of culture. This is the subcultured material. Next, we have different advantages in this particular plant tissue culture techniques. So availability of raw materials, aiming the two types, as mother as the daughter plants, and we can have these variations, nothing but hybrid varieties, and we can patent some rights upon this, and we can commercially exploit this particular products. And if you have identified any new component like new drug which is extracted from the plant basis or herbal basis or any other thing, so we can quote, if it's not done the experiment till now, so we can quote patent for that. Like as turmeric is a patent occupied by the US government, so where that is traditionally used in India, unfortunately, right. So they have got the patent right. They said that it has some kind of anti-inflammatory and antibiotic in natures, like that they have given. But we know that in the kitchen, when you have a small cuts or anything, we just add some kind of turmeric on that, right? So that clots the blood, and again it doesn't cause any kind of infections again. But the patent right was taken by the US government, right? We generally use that term here, that process everything here, and that is efficiently grown in the India. You know, Kerala is one particular region where it has 70 to 90 percentage of good curcumin content turmeric can be grown in the Kerala, and next to Telangana too. Some 70 to 80 percentage of good turmeric with good curcumin is cultivated in the Telangana region, and is as well as the Kerala, even some of the districts of AP again, right? Easy purification. So you can go with the easily purify the components without any time consuming tedious process, right? Next, modifying the chemical structures, even though knowing about its chemical components importance, like barbiturates, alkaloids, phenols, alkalines, everything can be. Even some kind of disease and desired products, some disease free plants, like good examples for disease with bt cotton genes, bt brinjal, where we are going to have complete eradication of this particular, when the gene is incorporated in that particular plant, like bacillus thuringiensis. When the insect or a worm come and preach the leaf material, suddenly it get paralyzed. Its jaws are going to be paralyzed, and it finally within 2 to 5 minutes the insect or worm is going to be died, right? So we can incorporate such kind of genes. We can have good gus gene expressions again, and followed by crop improvement for the healthy quality growth of crop improvement, like what you'll call golden rice, flavored savored tomato. Nowadays it is not that much of useful, and again having the good shelf lifetime of tomatoes, potatoes, everything. the tubers, not what may be, right? So these are the simple advantages of your plant tissue culture. Hope you all understood about the basic terminologies of plant tissue culture.